Okie doke. So, any specific questions so far on the heart? Everybody understand my favorite diagram in the whole book? You still can't figure it out? Uh-oh. All right, the question, I want you to click in. Who likes this question? A for yes and B, or who likes this diagram? A for yes and B for no. That's your clicker question today. A for yes and B for no. It's not a tough one, guys. Who doesn't have their clicker? Oi, that's not good. So our number's 39. Everybody who's got clicker has clicked in. Do you have your clicker?
Yes.
Click it. Good. Everybody clicked? 40's our number. So what should be your answer? There is no correct answer. Come on, guys. Yay is the correct answer. Okay, remember this diagram ties everything in, all the concepts in this chapter. Okay? So when you look at it, try and get it down here. Down here we're talking about valves open, atrioventricular valves. What's the aortic and pulmonary valves? What's another name for them? The semilunar valves. And what phase we're going through is down here. So if you follow it all the way up, you see all of these things going on during the first phase of the cardiac cycle. Yes? You see that during the first phase of the cardiac cycle, the semilunar valves are what? Closed. The atrioventricular valves are open. What about ventricular volume? Is it high or low? It's high because why? Because we're pushing everything out from the atria into the ventricles, big volume. How about the pressure in the atria versus the, what's that thing over there? The green thing. That's the aorta. So pressure's kind of low in the aorta for the aorta right now. How come? Yeah, nothing's getting pushed out it yet. Where's pressure highest? In the aorta, yes? You see how everything ties together? Okay, if we follow that up again, during this stage, what's happening? The atria are depolarizing. We see that in what? The P wave, yes. So what's there not? I don't get what there's not to get. You see how everything ties together? Okay, what happens when we go into the second phase? Who's going to close? Doors slam shut. There's your first heart sound. Yes? And what's going to start happening? Contraction of the ventricles. So what's going to happen to the volume? It's going to start to go down because what am I doing? I'm shooting that stuff out. Out either what or what? If I'm talking about the right, I'm shooting stuff to where? The lungs via the pulmonary what? Artery. Or if I'm talking about the left, I'm shooting things out the aorta to the what? To the system. Yes? So the volume goes down in the ventricles. What happens to the pressure in the aorta? That green line goes up. And then when I start to relax, it goes down a little bit. Yes? It's called the dicrotic notch. What happens when the pressure starts to go down and the semilunar valve slams shut? Second heart sound, yes? QRS happens in here, yes? What else happens when the ventricles contract? You can't see it in QRS, but that's happening also. The atria are repolarizing. And what's T? Keep going. Of the ventricles. Yes? The blue line is the left ventricle. Ventricle. Well, you know, when we look at volumes leaving the heart, it's usually volumes to the system. When we talk about stroke volume, we're talking about the volume that leaves the left ventricle. And that's why it's in the chart, not the right. But you still have the volume leaving the right ventricle too. That's going to where? Yeah. So you have the left atrium, volume going where? Into the where? No, left atrium. Dumping volume into left ventricle. Okay? Does it make a little bit more sense now? Yes? If your answer is still no, what do you have to do? Study. Yeah. Yep. If you don't quite get it after I explain it, then you got to go get it. And if you still don't get it, what are you going to do? Go find somebody that can explain it to you. You know where I live, don't you? Where do I live? Not at home in my house, in my office here at school. You can't come to my house. No. Where's my office? It's second semester with me. You should know where my office is by now. Jalbert 525. Right? You've got to figure it out. I passed anatomy. I'm good. Yeah? Just because you passed, it doesn't mean you. I think that was a diss. All right, so come seek knowledge, my friends. Use that cranium. Yes? The cerebral cortex is massive. Lots of files up there. Fill them. Fill the files with knowledge. Okay? Some stuff doesn't come automatically and immediately. Yes? That's life, my friends. When it doesn't come automatically and immediately, you have to go make it come. You have to go study. You have to go repeat. Or go find somebody that can help you understand a concept. Okay? Don't just sit back and go, I don't get it. Because guess what? You're not going to pass the class. Right? And then you're going to cry. And I hate it when you cry. So no crying. So bug me, bug somebody. Go figure it out. Okay? I'm recording all this too. So there. All right? So that's the cardiac cycle. What's this? Of course you should know that. You just had a test on that. It's a second messenger system. Very good. In this particular second messenger system, it happens to be a cyclic AMP second messenger system. And you know all about that because that was one of the essay questions on your exam. So in this particular second messenger system, we have a hormone called norepinephrine. And that is going to increase the contractility via a cyclic AMP second messenger system. Cause the contraction to be stronger in cells. That's the change in this particular second messenger system. Now, contraction strength is going to play a role in something called cardiac output and stroke volume. Now, cardiac output, what is that? It's the amount of blood pushed out of the heart during any given cardiac cycle. And typically, and we use an average here of about 70 milliliters per cardiac cycle. Now that is not a given number. It's different for everyone, isn't it? Sometimes it's higher, sometimes it's lower. So stroke volume can be affected by several different things. How elastic your heart is. So stroke volume is regulated by, and this begins on page 684 in your textbook, the degree of stretchability of your heart muscle. So when they talk about preload, degree of stretch of heart muscle, they talk about something called Frank Starling's law of the heart. Don't worry about that. But if the muscle is not strong, if the muscle is damaged, stroke volume is going to be affected. It is therefore going to affect cardiac output. So contractility of the muscle is going to affect cardiac output, how much blood you put out during any given cardiac cycle. They talk about afterload as well. So who regulates heart rate? The central nervous system, the autonomic nervous system, and also who else? Sinoatrial node, atrioventricular node. The autorhythmic cells, right? All those guys are going to work together to help regulate heart rate. So the autonomic nervous system, and this happens to be an example of some hormonal regulation of cardiac output and contractility of the heart in this, oh, wait a minute, I've switched it. In figure 18.21 with norepinephrine. Who makes epinephrine? The who? The adrenal glands, right? So hormones can regulate contractility of the heart. Not only can hormones like epinephrine and norepinephrine regulate heart rate or contractility, but hormones from the thyroid gland can do that too. Things like thyroxine. What's that? T4. So that can also have an effect on heart rate and contractility. And then ion concentration. Sometimes when our patients have problems with dehydration, they also might run into problems with heart rate regulation and regulating heartbeat and contractility. Potassium, for example, when it goes way high, it can kill you. If you want to kill somebody, if you want to kill, you know, one of the ways is to really mess up your ion concentrations. To load potassium can stop your heart from beating. No, you'd have to eat one heck of a lot of bananas to get that much potassium. But ion imbalances can be a big problem for electrical events in your body, including heart rate and heart contractility. So one of the things you have to be careful of is not becoming dehydrated. And one of the things you have to be careful of is putting things into your body that have a high ion concentration. One thing in particular that drives me crazy because it's sold right in the cafeteria to those of you who are going to drink the beverage and then sit on your keister and do nothing. Do you know which beverage I'm talking about? Gatorade. Gatorade is an awesome drink. For who? Somebody who's sweating profusely and running around like crazy. Not somebody who is sitting on the couch watching SpongeBob. Because it affects your ion concentrations drastically. It's meant to replenish lost ions. So don't drink Gatorade unless you are exercising and losing fluids and electrolytes. It's not good for you. And plus there's a whole lot of sugar in there and you don't need that anyway. So ion concentrations are very important. Some of the imbalances that can result from disruption of ion concentrations. Calcium is another very important ion. Of course, everybody went in and looked at that interactive physiology to help them study for this quiz. So you know how important calcium is to the whole autorhythmic cell, right?
I tried, but my laptop wouldn't let me.
Your laptop wouldn't let you? Oh, booger. Go into mastering. You don't need the CD to go into interactive physiology. Did you know that? And if you can't see it on your computer, you might have to do some free downloading. Does everybody know that? You might be missing a plugin. They're free. Go check, see if you can download it. It's right within the program. Okay? Use it. It's going to be very helpful. Age is going to affect heart rate and contractility. The amount of exercise you get or the heart muscle gets is going to affect contractility as well. So potassium is a biggie, calcium is a biggie. What happens when heart rate goes way too low? That's called tachycardia. What's that? Excuse me, bradycardia. Heart low. Heart high is tachycardia. If I'm going way low, bradycardia, what's that? Who cares? What's the difference? What's that going to do to my cardiac output? It's going to decrease it quite a bit. Is that good for me, getting all my nutrients and waste and oxygen and carbon dioxide around? It's going to slow way down. How about if I have tachycardia? When my heart goes too fast. Do you think my heart is going to be as efficient as it should be and contracting and pushing out volumes when it's going way too fast? No. So they are described under homeostatic imbalances as well. Other imbalances, other things that can affect how much blood your heart puts out in any given cardiac cycle are described on page 687. Coronary and yes, it's TH, atherosclerosis. What's that? Yeah, what's clogging up the arteries in atherosclerosis? Lipids. Okay. So, if I have a problem carrying around lipids in my circulatory system, and there are wonderful little protein carriers that are going to help me do that. Anybody ever hear of the good cholesterol and bad cholesterols? HDL, LDL, VLDL. Okay. Those are not cholesterol. They're cholesterol what? They're carriers of cholesterol. We call them lipoproteins. Because in order to get those lipids around in the circulatory system, they have to have some sort of water-soluble carrier to get them around. Now, HDL is very strong, high-density lipoprotein, and puts cholesterol where it belongs. Precursors to making HDL, the low-density lipoproteins, LDL, and very low-density lipoprotein, VLDL, they carry cholesterol too, but they're not as efficient at doing it. Sometimes they drop it places where it doesn't belong, like in the lining of your arteries. And things might build up in there and block up tubes. That can be a big problem. Yes? Yes. If we block a coronary artery or coronary vein, then we're blocking the food supply to everybody who's past that blockage. What's going to happen to that portion of the muscle? It's going to die. It doesn't get any food. What's that called? Well, when the heart muscle dies. And you have a heart attack. Myocardial infarction. Infarction. You have a question?
Yeah. When you initially have like a heart attack, it's pain in the shoulders.
It's called referred pain.
Yeah, I know that. But why? I guess what's up?
It's because of the way your nervous system is set up.
What are you, I know that you said what exactly?
When tissue dies, when tissue dies, or when you cut tissue and tissue dies, it fires off pain receptors. So you are, because of dying tissue, firing off groups of pain receptors that happen to make you feel as if you're having pain in those areas because of the different pathways in the peripheral nervous system.
So the tissue dies immediately, not like
Oh, gradually, yes. Yeah. It's going to die fairly quickly if I have a clot floating around and it plugs it up. Atherosclerosis, typically the tubes get thinner and thinner and thinner as far as volume being able to go through them. So that's going to slowly happen. But yes, there are situations where I can block that quickly. And what kind of clot floating around, what do we call those? An embolus. So if we have an embolus that ends up in a coronary artery, then we can have that happen fairly quickly. So, pressure in the system. When it's too high, what do I do? When I continually apply a very high pressure on a bunch of tubes, I'm going to wear those tubes out faster, aren't I? So normally pressure in the aorta during diastole. What's diastole? Well, kind of on the right track. What's systole? Contraction, depolarization. Another word for systole. What's diastole? Relaxation or repolarization. So it should be at 80. 120 over 80. Yes? That's blood pressure. We're going to talk about it in the next chapter. So the bottom number, the diastolic pressure in the system, should be about 80 millimeters of mercury. So when it gets high blood pressure, usually that's the one that we're most concerned with, because that's the lowest pressure in the system. If the lowest pressure in the system is really, really high, then you're applying quite a pressure to your tubes. Yes? That can cause problems with contractility and pushing out volume, and again affect cardiac output. Multiple myocardial infarctions, hopefully that's self-explanatory. If the tissue dies, it's not going to be able to contract and relax and push out a volume as efficiently. So that's going to affect cardiac output. And what's a cardiomyopathy? Ever have an elastic that's been around for a very long time? It doesn't snap, but yeah, it gets way stretchy, and it never really goes back to the way it was when it was nice and brand new. Yes? So think of your heart muscle that way. When you look at the definition of a cardiomyopathy, it states right in there, the cause of this condition in which the ventricles stretch and become flabby. So they don't contract and relax the way they did when they were nice and brand new. So what's that going to do to cardiac output? So if I can't do this anymore, I can only do this. What's going to happen to cardiac output? It's going to go down because what's going to go down? Stroke volume. Okay? And the pressure in the system will go down too, you're right. So understand some of the different imbalances of cardiac output. Different developmental aspects of the heart. Don't worry about it. This diagram ties together some of the different things that affect cardiac output. So, exercise, skeletal muscle, and respiratory pump. We're going to talk about that in the next chapter. What's that do to cardiac output? I have to go get my Hermit's in the bookstore. Yeah. Heart rate. Decreased heart rate allows more time for ventricular filling. And that's going to increase cardiac output as well. Blood-borne epinephrine, thyroxine, and excess calcium. What's that going to do to contractility of the heart? Increase it. Is that going to increase or decrease cardiac output? It's going to increase, because I can do what now? I can push harder and pump out more. Exercise, fright, anxiety, increases sympathetic activity, which increases contractility, which eventually does what? Follow all the arrows down. Yep, increases cardiac output. An increase in heart rate. And just do the math. If I increase the number of beats per minute, I'm going to increase what? Cardiac output. That's if stroke volume stays fairly consistent as well. Okay? So these are some of the different things that can affect cardiac output. Are we good? All right, that's it for chapter 18. Let's hit chapter 19. Any questions on... I don't know. Where did my little... Oh, there it is. So, what's chapter 19? Blood vessels. And what do we call those blood vessels that we're interested in talking about? Arteries, capillaries, and veins. Now remember, this is a closed system. Everybody understands what that means by now, right? What does it mean? Yeah, there's no end. So I have a pump. We already learned about that, the heart, connected to a bunch of tubes. There's no openings. The tubes are in a complete circuit. Yes? I have no idea. I think the pipes all kind of feed around. I think it's a closed system, isn't it? Yeah. But like forced hot, not forced hot air, forced hot water is a closed system. Is that good? Think of the pump and the tubes. Okay? And I think yeah, there is one. We'll get there in a second. All right. So we're talking about the blood vessels now, the carriers of blood. They're coming from the pump, and we've already heard their names, arteries and veins. There's ones that connect the arteries and veins. Those are called the capillaries. And this is where the business end of the exchange in circulatory system happens, in the capillary beds. So when you finish your lab exams this week, everybody take your lab exams when you're supposed to. What if you can't go to your lab and take a lab exam? Go to another section. Yes? Take your lab exams this week. Arteries and veins are a little bit different. And it has a lot to do with the fact that arteries are under much more pressure than veins are. Arteries carry blood what? Away from the pump. Away from the pump. And the pump pushes blood into them. Makes sense that they're under more pressure? And then in that closed circuit, veins are going to do what? Bring blood back to the heart for another push. So that implies that the pressure is going to be a little bit less, yes? Because as we travel through the tubes, the pressure in the system is going to go down. So if we look at them under the microscope, we can see the difference between an artery and a vein. The artery tends to be more wide open, but this is, don't be fooled by this histology. Sometimes when you look at tissue under the microscope, your arteries can be sort of collapsed on themselves as well, only because they're not under pressure anymore. But arteries are thicker walled than veins of the same size. Veins don't have as thick a wall because they don't have to be under so much pressure and they don't have to absorb as much pressure. So you should understand then the structure of the artery versus the structure of the vein. What we're going to see in the artery, the red stuff on the left side of your screen, is that the walls are much thicker. The walls of arteries and veins are called tunics or tunica. And how many different tunics or tunica make up arteries and veins? 3 of them. We have one on the outside called the tunica what? Externa. And what's it made of? Like connective tissue material. Lots of collagen fibers, very thick. It's kind of like wrapping paper, right? Then underneath that we have the tunica media. And the tunica media is the tunic in the middle, bless you, made of what? Muscle. Muscle for what? What do we need muscles and arteries and veins for? To squeeze blood through. Yeah, for vasoconstriction and vasodilation. Very good. Now wait a minute. There's an extra little layer that looks like Swiss cheese that we find in the artery that's not in the vein. What's it called? The external elastic lamina. What's that for? More pressure, right? If I'm under more pressure, I need to be able to do what? Stretch more. So we're going to find extra layers in the arteries that we don't find in the veins. We'll find one in between the tunica media and tunica externa. It's called the elastic lamina, or external in this case. We find it in the arteries. It's going to give them a little bit more stretch. Both of them have smooth muscle in their tunica media, but whose tends to be thicker? Arteries. Why? They're under more pressure, exactly. Then we have the tunica intima, the internal tunic, which is basically endothelium. What's endothelium? Somebody said it way back in there. Simple, squamous, epithelial cells, lining things, covering things, yes? And that's what that is. So that internal layer is an epithelial layer lining the internal tube. And again, we have connective tissue in both arteries and veins that are kind of holding everybody together. And then we see that other layer of Swiss cheese, the internal elastic lamina that we find in the artery and we don't find in the vein. How come? More stretchiness. Yes? What's that? A valve? Do we have valves in arteries? No. There's valves in the heart. But in arteries, there's no valves. What the heck is a valve doing in a vein? Blood pumps from the heart. Yes? Big push. Going out the arteries, everybody's getting it. Even my toes.
Slows down in the veins, so there's more chance of
Backflow, exactly. So when I get to my toes, the tubes are going to get smaller and smaller and smaller and smaller. Question?
Yes. On the pictures on the artery and vein, the artery is thicker than a vein, but it looks like it's thinner
Yes. And I'll talk about that in 1 minute. So, push out from the heart, travel through the arteries, get to my toes. Tubes are going to get smaller and smaller and smaller and smaller until we get into what? The capillary beds. What's going to happen down in the toe region? Exchange of gases that I picked up from the respiratory system, and then I'm going to throw my waste gases in, and I'm going to throw in some waste from the tissues. Now I'm going to continue back to who? Back to the heart. So in the capillary beds, we have the switch between artery and vein, kind of in this gray area where the whole exchange thing takes place. Okay, I'm not getting any more pushing. The only pushing I'm getting is from everybody behind me at this point, right? And now I have to go back to where? From my toes. I have to go back to the heart. Do I have tons and tons of pressure? No. I'm only going to get pushed from behind. So if I took everybody in this classroom and I lined you up, right up against each other along this wall, I would be the heart at the end and push the first person. By the time the person at the end felt that push, would it be nearly as strong? Okay, so you get that? So we're going to have problems with pressure, not enough pressure. So we also have help. Help in the venous system from valves that'll prevent backflow. And we're also going to have help from different surrounding muscles for some of our major veins that are feeding back to the heart. Every time you breathe in and out, what happens? Gas exchange. But think about your thoracic cavity. It expands and pushes up against everything that's touching it, including some major veins, and gives a little what to them? A little squeeze to help get blood back to the heart. That also happens down in your legs. And if you've been standing at attention for a very long time, what might happen to you? You might pass out. How come? Yeah, you're not getting that blood from your lower extremities back. So when soldiers stand at attention, what are they told to do? Slightly bend the knees, or just pick up your heels, or maybe your heels a little bit. Yes? So you can contract those muscles. Those are called muscular pumps. They're in your legs, they're in your thoracic cavity, they're in your abdominal pelvic cavity. And the muscles are just basically squeezing the tubes, the veins, to get volume back to the heart. Help give it a little bit more push. Did you have a question? You figured it out? Good. Okay? So when we talk about the blood vessels and the structure, you should understand that the arteries are thicker walled. Somebody said something about the lumen. What's a lumen? The space on the inside. If I have to water my bushes with my garden hose, and I don't have one of those fancy schmancy little handles, all I have is my garden hose, and I have to water my bushes way over there, how do I do it? I put my thumb over it. What's that do to the lumen of my garden hose? It makes it smaller. The pressure from the house where I put the water on is still coming at the same pressure, right? What's going to happen to the water distance? It's going to go further because the lumen is what? Smaller. So things are going to move faster through the arteries than they do through the veins. The veins have larger what? Lumens. So we'll talk about that when we talk about resistance in the system and pressure in the system. So the capillary beds, basically, once we hit that point, once we hit this point in the capillary beds, this is pretty much all we have. A single layer of endothelial cells and a little tiny wrapper of basement membrane to hold everybody together. Now do you see why exchange can take place? Can exchange take place through this wall? No, too thick, right? So it has to happen in the capillary bed. Same with the veins as well. So they are just transporters of blood to get them to where they're going. So when we talk about the system itself, we see it's a closed system. The heart is the pump. The arteries are going to take things away from the pump. The veins are going to bring things back to the pump. An exchange is going to happen in the in-between connector between arteries and veins called capillaries. What the heck are those green things in there? The lymphatic vessels. We're going to talk about the lymphatic system in the next chapter. But lymphatic vessels are also very important to fluid circulation through the body. Exchange is going to happen down in the capillary beds. Not only wastes, oxygen, carbon dioxide, all of that good stuff, but fluids are going to leave the circulatory system and enter the circulatory system in the capillary beds as well. One of the problems with the pressure in the system is that a little bit too much with respect to fluid leaves in the capillary beds, then can be reabsorbed before everything gets pushed along. So who's going to help me pick up the slack with that excess tissue fluid? The lymphatic system. That, when we discuss the lymphatic system, is a series of tubes very similar to the venous system, attached to things called lymph nodes and lymphatic tissue, which is also very important for keeping us safe from foreign nasty invaders that might get into our fluids. Eventually that excess tissue fluid for the circulatory system is going to dump back where? Just before everybody gets back to the heart through the vena cavas, up and around this region, and again we'll talk about it next chapter, we're going to dump that lymphatic fluid back into the circulatory system. So the lymph vessels, one of its many jobs, is to help regulate fluid balance in the body as well. It's going to help the circulatory system. It's kind of a way indirect route, but yes. So there's a bunch of different types of tubes that we'll find throughout, and this is a diagram that talks about some of the arteries. And what we're going to see, the further we get away from the heart, the histology of the artery is going to change a little bit. The arteries that are very, very close to the heart, and this is a nice diagram because it ties everything together again. Love the diagrams. Look and see how everything works together. In an elastic artery, which is closest to who? The heart. Why do you think elastic artery is closest to the heart? That's where I'm getting the push. That's where I have to be able to stretch the most and compensate the most. So we see that the different layers that we just discussed, endothelium, always going to stay the same, single layer lining. But what happens to the elastic layer in the elastic arteries? It's much thicker. Why? It needs to be able to stretch more and absorb the pressure that's being sent from the pump. Smooth muscle layer is also fairly thick, but not as thick as the next group of arteries, which we call what? Muscular arteries. Why do we call them muscular arteries? Yeah, they have the thickest of muscle layers. So, another name for the elastic arteries are the conducting arteries, closest to the heart, absorbing a lot of that pressure from the pump. The muscular arteries are also called what? Distributing arteries. So now, once everything leaves the heart, what do I have to do now? Yeah, bring it around to everybody that needs it. Send it to places. Send it to my heart. Send it to my brain. Send it to my abdominal pelvic cavity, my legs, my arms. So the muscular arteries are going to help push that blood with its muscle through the system to where it needs to go. Again, they're also called distributing arteries. As we start to get smaller and smaller, what is going to happen to the name of my arteries? Who's after the muscular arteries? They're called arterioles. So think of this as a tree. The aorta would be the trunk of the tree. And when I finally get the goodies to where they need to go, the leaves, what's going to happen to the size of that trunk? It gets smaller and smaller and smaller. Thinner and thinner and thinner, so we can have exchange happen. So as we get smaller, we go to what we call the arterioles. And that's where we're kind of distributing blood to very specific places in the body. Who's next? Capillaries. Look what happened. We went from muscle and elastic and then that connective tissue covering on all of those guys. And then we hit the capillary beds, and what do we got? One cell layer thick. That nice endothelium. Very, very, very little bit of connective tissue holding everybody together. So, since it's a continuous system and there's no holes in the system, after we pass the capillary beds, where do we go? We then go into the venous system. It's a continuous tube, but now I dropped off the goodies in the capillary system, and I tend to have deoxygenated blood now in the venous portion. Going back where? Now I have to go back to the heart. Why do I have to go back to the heart? A, I need another push because I'm losing pressure and I'm losing it fast. B, I have to drop off the waste gases, carbon dioxide, to who? To the lungs. So I'm going to go back to the right side of the heart, take a trip to the lungs, and then come back to the heart for another what? Another push. Now, capillary beds are of different kinds. And in your textbook, starting on page 699, we start talking about some of the different types of capillaries. Some of them are what we call continuous capillaries. And why do you think they call them continuous capillaries? Yeah, the cells, the endothelial cells are all nice and beside each other. There's not a lot of break. If I'm going to exchange something from the inside of those tubes to the outside of the tubes, who's going to help me do that? If I want to get something from blood, and that's where the big honkin' red blood cell is, if I want to get something from the plasma, for example, from a continuous capillary into the tissue that's outside that capillary bed, how do you think it's going to get in and out? Can cells help me do this? Through what process? Last semester. Stuff I got to remember. How about? Diapedesis, that's squeezing through the spaces. Ah! Someone got it! Endo and exocytosis. A cell can suck something in and then spit it out on the other end, right? But that's going to be very selective, isn't it? It's going to take me some time to do that. So continuous capillaries are the least permeable of the capillaries. Skin, muscle, brain. Remember when we talked about the central nervous system, we talked about something called the blood-brain barrier, and we talked about those lovely little ependymal cells that were going to make sure only certain things left the plasma and got into my cerebrospinal fluid. Remember those guys? What kind of capillary beds? Continuous capillary beds. Very selective, takes its time doing what it needs to do. Yes? No, I remember I have to supply the brain with all the goodies, right? So what do I have a bunch of in my brain? Capillary beds. All over your whole entire body. Capillary beds. Okay? So these are places where I'm going to find continuous capillary beds. The next ones we call fenestrated capillaries. I'm going to teach you some Italian. What is window in Italian? Finestra. There you go. Italian lesson. Now, the cells of the endothelial cells of a fenestrated capillary bed have what in them? Little windows or fenestrations. Can things move in and out a little bit faster here? Yeah. So because of those holes or fenestrations in the cells, we can see things move in and out of the circulatory system a little bit more quickly. What happens in the kidneys? Blood filtration, somebody said. Filtering what? Yeah, we get rid of waste products that the blood is carrying around, because that's one of the jobs of blood, right? And we put it out in form what? Urine. In the kidneys. Where else? Small intestine, what happens there? Anybody eat anything today? I had a small piece of chocolate. No, I had lunch too. It was one of those little Andes candies. I enjoy those. Yeah, the mint ones. Gotta have a little chocolate. It's Valentine's Day, right? So, yeah, and dark chocolate is very high in antioxidants. Very good for you. So, I eat it, then what do I have to do with it? I break it down, small, 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 and then what do I do with it? Absorb! Let's get to the absorb part! Goodness gracious! So where do I absorb those molecules into? The circulatory system. From where do I have most of the absorption happening? Small intestine. Yes? We getting the point? No, I don't think you are. So, small intestine, kidneys, places where exchange can happen a little bit faster because of the fenestrated capillary beds. Oh, whoa, what the heck's going on here? All sorts of problems with these guys. Big, huge, massive holes. What kind of capillary is this? No fenestrations anymore. Sinuses, big, huge spaces. Where in goodness name would we find things like this with big holes in them? Why would I find them in the bone marrow? Oh, I just had a test on blood. Blood is created in the bone marrow, blood cells that will circulate just like our lovely little friend, the erythrocyte. And how does it get from the bone marrow into the circulatory system? What the heck did I just do? There you go. What did I do? Was I leaning? I'm using my hands too. So, places like bone marrow where big cells have to squeeze out from the bone marrow and go into the circulatory system. How do you think they get in there? Through those big honking spaces in the sinusoidal capillary beds. What do I do in the liver and the spleen? Why would I have big honking holes there? I get rid of old red blood cells. I recycle them. Where do I recycle some of them? That whole bilirubin, urea thing in the liver. Who else does that for me? Yep. So bigger things have to pass in and out of the capillary beds, and that's where we find sinusoidal capillaries. The most permeable, lots of things flow in and out here. So, the capillary beds themselves. So you have to know the difference between the capillaries and what they do for a living. In the capillary beds, this is where we're going to have exchange. And again, when we look at the picture, when we look at the diagram, we see kind of the gray area. And why do we color it gray? Yeah, that's where we kind of have the exchange, and we go from artery to vein. So, the terminal arterioles, starting to get very, very small near the end of the artery tubes, will feed into what we call the meta-arterioles, starting to get very, very small to the business end of the arteries. And that's called the true capillary bed. So that gray area is where exchange is going to take place. Now, any given capillary bed can have many, many different branches on it. Because what do I have to do? In the capillary beds, I'd have to have exchange. Exchange for who? Yeah, everybody, right? So I have to make sure I distribute as much as I possibly can in any given area all of the goodies in the true capillary beds. There's tiny little muscles surrounding all of those pre-capillary regions before the true capillary bed. And those little muscles, we've heard this word before, are called what muscles? Sphincter muscles. Sphincter muscles do what? Contract and squeeze off the tube and dilate and open the tube. So sometimes when I need to move blood fairly quickly, I might close off some of those sphincter muscles, and we see that down in this region, so we can bypass a certain area and get blood quickly through the capillary beds. At other times when an exchange needs to happen, those sphincter muscles will open up and we'll have blood flow through all of the different branches in a capillary bed. So the true capillaries is where the exchange part happens. And then we are going to feed that closed system into what? After the capillaries, what do we call it? We don't call those tubes arteries anymore or arterioles. We call them veins. Beginning ones are called venules, the small ones. Oh, that's my phone. Sorry. So, they're called veins or venules. I'm going to go back to this diagram. So, after we pass the capillaries, we're going to go into the venous system. The beginning small little veins are called venules. We start to see what's happening to the walls. They're starting to thicken up. We're starting to get muscle. We're starting to get connective tissue. As we keep flowing along, we're going to thicken up even more. When we turn into a vein now, we can see that we have a little bit of elastic tissue, much more muscle, and much more what? That connective tissue material. And then where are we going to go from there? Back to the heart. Correct. So in the venous system, venules are the beginning. Post-capillary venules, after the capillary bed, we're going to start into the venous system. Then we're going to turn into veins, and eventually we're going to dump back into the heart. Much of your blood volume is hanging out in the venous end of the tubes. How come? Think of me pushing you guys. Yeah, because there's a lower pressure. You're going to hang out there a little bit longer because the pressure is low and not a lot of push is happening from behind. So it's going to take you a little bit longer. Blood reservoirs, because of up to 65% of blood's volume supply is found in the veins at any given time. So most of the blood, 65% of your blood, is trying to get back to the pump in the venous system. So when we have a look at the breakdown, we see this diagram on page 701 in your textbook, relative proportion of blood volume throughout the cardiovascular system. We see much of it is in the venous portion of the tubes. Capillary beds about 5%, the heart only 8%, pulmonary blood 12%, that's the stuff going to the lungs to get rid of what? Carbon dioxide pickup, oxygen, and things move fairly quickly through the arteries because they're under what? High pressure, so only 15% of blood is traveling through the arteries at any given time. Remember, where's the business end happening? The capillary beds. What's an anastomosis? Anastomosis. A-N-A-S-T-O-M-O-S-E-S. Yeah, we have to have quite a vast array of tubes to make sure everybody gets what they need at any given time. So it's connections or branches, an anastomosis, off tubes. So we have different channels or different pathways to a given area. I'll give an example. If I have to get to tissue under here, how many ways can I get to it with this series of tubes? Well, I can get to it this way, and I can get to it this way, and I can get to it this way. Yes? So there has to be several different ways to get to an area, and that's what they talk about when they talk about an anastomosis. Different connections, different offshoots from areas. Some arteries do not anastomose. They develop poor circulation, and some do. When you gain adipose tissue, what do you also gain? Yeah, that's fat, that's adipose tissue. But what else do you also gain? More branches off capillary beds. Why? Everybody's got to get fed, right? So I have more cells, I have to make more branches or collaterals off a given region so I can supply those cells with the nutrients and get rid of wastes from them as well.
So when you gain adipose tissue, they create it, does that create more stress on the whole system itself?
Correct. Yep, because the tubes are longer. Yes?
So does that mean just the
You have more difficulty getting things back to the pump for a push.
does gaining more adipose tissue also mean that your body wants to make more red blood cells and stuff? Or is it just the same amount of red blood cells, more red blood cells?
Yep, more oxygen carrying capacity. Okay? So that's what they talk about under anastomosis. In your textbook on page 702 and 703, they give us a little look or closer look at atherosclerosis. Nasty! So what happens is a buildup, and this guy over here, the blue one, this one's in real trouble here, yes? What happened to this lumen? It's now this big. Yes? I don't know. It's a blood vessel. It's an artery. So do we understand why that's a problem? Getting things through those tubes? Okay, what else can we also increase risk of? Heart attack, stroke, dead tissue on the other end, but what else? When I start rumbling up blood as it moves through tubes, I might initiate a clotting cascade. So when I have vessels like this, I increase my risk of intrinsic coagulation. Yeah?
You said that arteries were very pressured, right?
Arteries are under more pressure, yes.
So has it ever happened where like an artery has been so clogged instead of having a heart attack or stroke or something like that can actually burst there?
Yep, all the time. What's that? What could that be called? An aneurysm. If it happens in the brain, a cerebrovascular accident. Yes? So it can weaken the vessels and cause them to burst for sure. That's called an aneurysm. Physiology of circulation is next. Is this a good place to stop? Oh, I was just getting so excited. Okay, we'll pick up with physiology of circulation on Thursday.
Did you count the clicker foot as a fail and pass?
What, hon?